My name is Zeng. I'll introduce the paper submitted to iMeta-omics. The title is Multi-omics Analysis of Gut Organ Axes Reveals the High-Altitude Adaptation in Tibetan Chicken. The Qinghai-Tibet Plateau has been described as the third pole of the Earth because of its extreme environmental conditions, such as low oxygen level, high UV radiation, and cold temperature, which are challenging for many species. One key aspect of animal adaptation to hypoxic environments is gut microbiota, which is closely associated with host health and environmental adaptation. Several studies have found significant differences between the intestinal bacteria of high and low-altitude humans or animals. However, the assembly mechanisms of gut microbiota in Tibetan chickens regarding low-altitude adaptation are still missing.
We performed a multi-omics analysis on 6 Chinese chicken populations based on geographic differences in altitude.
The main contents of this study are as follows. We measured 6 blood physiological indices among the different chicken populations. The concentrations of hemoglobin, mean erythrocyte hemoglobin content, mean corpuscular hemoglobin concentration and mean corpuscular volume in INTC were much higher than other group, while no significant difference was observed among plains populations. To investigate the effect of gut microbiota composition regarding high-altitude adaptation, we sequenced cecum content samples from 6 chicken populations. The results found that the Chow-1 and PD_Faith index showed obvious differences between the plateau and plains populations of Tibetan chicken. These results suggest a higher richness and phylogenetic diversity in gut microbiota of chicken populations from the plateau compared to those from lowland plains. We further applied unifrac distances to assess beta diversity of chicken cecal microbiota. Principal coordinate analysis showed a distinct separation among groups. The contributions of turnover were more important than nestedness. We further quantified the relative contributions of alpha and beta diversity to gamma diversity. The alpha diversity of inticulation contributed about 97% of the variation. In contrast, beta diversity showed the opposite trend. According to our taxonomic analyses, Firmicutes was the most abundant bacterial phylum. The relative abundances of proteobacteria and actinobacteriota were significantly lower in the INTC samples compared to those in the plains population samples. Among the studied chicken populations, only 21 bacterial amplicon sequence variants were shared. These outcomes further validate that differences in the local species pool were drivers of the interpopulation variations in chicken cecal microbiota. We used the BETI index to evaluate the gut microbiota assembly in different chicken populations. Median values of BETI were between -2 and 2, except for the LS group, which had a value slightly above 2. These values indicate that stochastic processes governed the assembly of a majority of the microbiota. There were still population-specific differences. We observed bacteroids were significantly enriched in the INTC group, while Limosilactobacillus and Lactobacillus were more abundant in XTC individuals. The predicted functions indicated a higher abundance involved in cellular processes in the INTC group, and more abundant functions related to human diseases in the XTC group. The null model analysis indicated the gut microbiota assembly have changed from a stochastic-driven state in the INTC group to a deterministic-driven state in the XTC population. We investigated the gut metabolome to explore the potential mechanisms behind the plateau adaptability. Principal coordinate analysis showed separate clusters of different populations. Differentially abundant metabolites in INTC versus XTC were significantly enriched in pathways of purine metabolism, ISO, flavonoid biosynthesis, and biosynthesis of unsaturated fatty acids. In addition, we compared the transcriptomes of the lung, heart, and liver tissues among the different populations to further investigate the plateau adaptation. We found natural killer cell-mediated cytotoxicity, and cytokine-cytokine receptor interaction may be crucial for high-altitude adaptation. It is noteworthy that we identified ASV 53, 9, 65, 67, and 131 as potential key ASVs in all 3 investigated axes of the Tibetan chicken. In summary, we carried out a multi-omics study to explore the environmental adaptation mechanisms of Tibetan chickens. We found Tibetan chickens exhibit distinct gut microbial communities shaped by high-altitude conditions with higher functional redundancy and stochastic dominance and have a more effective fatty acid degradation capacity. Meanwhile, Firmicutes as regulators of host environmental adaptation, which contribute valuable insights into the adaptation of chicken gut microbiota to high-altitude environments.